Thanks very much for that kind introduction and for inviting me here today. Because I live in Halifax and trained in Boston and Toronto, I can support whatever sports team happens to be winning. So I'm going to talk to you a bit about our work using zebrafish to model Li-Fraumeni syndrome. And you've already had a couple of talks that have introduced the zebrafish to you, and my job today is really to further show you how the zebrafish is really a valuable model to understand human disease, and specifically Li-Fraumeni syndrome, but also other cancer predisposition syndromes. Now, there's a number of different animal models that people use in cancer research. You've heard about mice, you're hearing about zebrafish, and there's members in the audience like Brent Derry that use C. elegans, which are worms, and flies. And these models have a variety of opportunities, advantages, challenges, not the least of which is cost. But my job today is to show you how we think the zebrafish sits evolutionarily really at a critical point in being vertebrates, so they're closer to us than flies and worms, but not being mammals like mice that have some of the advantages you've already seen in terms of our ability to have large numbers of fish that we can keep relatively inexpensively, and the power of imaging. And what I'm going to emphasize in my talk today is the power of genetic manipulation and how we're able to use that to make really accurate models of Li-Fraumeni syndrome in zebrafish. So why would we want to do this in zebrafish? You've already heard some of the advantages. Zebrafish undergo rapid development. I'm going to play this movie that takes you through the development of a zebrafish embryo for the first 22 hours of life. This is the zygote, the cell that's split, and you can see that the zebrafish rapidly undergoes rapid cell division, forming a multicellular organism. There's high degree of genetic conservation, which is really important so that mutations in the zebrafish match mutations in humans. I'm going to emphasize our ability with genome editing technology to be able to specifically knock out and knock in genes that are relevant, particularly the power of CRISPR that has really revolutionized what we're able to do in the zebrafish. Once we've made these models, we can then apply high-throughput assays to look at specific phenotypes in these fish, particularly apoptosis or lack of apoptosis, which is really a hallmark in Li-Fraumeni syndrome. And then, as Dave alluded to, we've been using the fish to do chemical screens. In this case, we're doing this in larvae, in the baby fish, not in the adults. And what we're really focusing on is the ability to find drugs that can prevent the formation of tumors. So you've seen how zebrafish can be helpful in finding drugs that treat tumors, but really, particularly in in cancer predisposition syndromes like Li-Fraumeni syndrome, wouldn't it be great if we could actually prevent the tumor before it develops? So, importantly, zebrafish have high conservation. You've already heard how they're conserved really on the gene level and on the chromosome level. But it's also important to recognize as vertebrates that they're conserved at the organ level. And this is really important when we think about doing drug trials in larvae. So the conservations, even in the baby fish where they have hearts and kidneys, gills that function like lungs, all of these organ systems, livers that are very similar to humans, means they metabolize drugs very similarly to the way humans do, but also that they develop similar side effects and toxicities. And so I'm going to mention how we can also use the fish not only to find new drugs, but also safer drugs. So how did I get involved with Li-Fraumeni syndrome? I'm a pediatric oncologist, and I had the opportunity a number of years ago to be recruited by David, who's been a wonderful mentor to me for many, many years, as part of a growing multidisciplinary team on a project funded by the Terry Fox Research Institute. And this is really using all of these different modalities in terms of genomics, imaging, and more recently, circulating tumor DNA, to work together with this group to use zebrafish as a tool to test in vivo many of the genetic alterations that are being found by David and Adam and other members of this team, and then working together with people like Anita and Ron to actually test therapeutics in these models and bring them back to clinic. So as you've already heard, there are a number of different zebrafish models of P53 mutations. You've heard about the 214K mutation from Dr. Lane. You've heard about the P53 knockout from Dr. Langenau. And where we're contributing to this literature is making these specific Li-Fraumeni mutations. Because as you heard from Dr. Lane, knocking out P53 in the zebrafish, which is what has been done up until now, is of value, but is not necessarily what happens with every P53 mutation, and certainly not in Li-Fraumeni syndrome, because some of these mutations actually are gain-of-function mutations, not loss of the gene. And so if we really want to be able to model this disease in zebrafish, we need to be able to introduce the specific mutations in zebrafish that are found in Li-Fraumeni syndrome. And I'm going to show you how we've done that. Importantly, once we've made these mutations, we have assays that we can use to screen for phenotypes and specifically to look for loss of apoptosis. And we can use something called a tunnel assay or acridine orange. And apoptosis is cell death. So one of the main roles of P53 is to be able to control a cell living or dying when it's exposed to DNA damage, whether that's radiation, chemotherapy, or other DNA damaging agents. And when P53 is lost, those cells don't die. They can accumulate other mutations and develop cancers. And so that's really a hallmark of loss of P53 function and something that we're taking advantage of in our fish models. So the way we've been doing this is taking advantage of this technology called CRISPR, which many of you have heard about and has already been mentioned today. And CRISPR really functions as a cut and paste tool, the way you would in a word processor, cut out a word or a letter and stick it someplace else. And that's really what CRISPR allows us to do. It allows us to take the specific mutations found in Li-Fraumeni syndrome and introduce them into the same spot in the zebrafish genome where they're found in the human genome. And again, because we have a high degree of conservation between the P53 gene in zebrafish and humans, we can find those specific residues and make those specific mutations. And so we've been working specifically on this 175 mutation and this 217 mutation. And this cartoon here just shows you a schematic of how CRISPR-Cas9 works. It takes advantage of this protein called Cas9, which is like the molecular scissors that cuts, and you develop these guide RNAs that specifically localize on that place in the genome, telling that Cas9 where to cut, and then you have another piece of DNA called an oligo that contains your mutation, and it inserts that mutation at that cut site. And using that approach, we've been making these Li-Fraumeni fish, or these CRISPR Li-Fraumeni fish. And this is an example of how we've done this with the 143 mutation and the 217 mutation, and this work has really been championed by Sergei Prekozic, who's a research associate in my group. And here he's shown you the strategy for how we knock these in. With CRISPRs, they have these PAM mutations, which is what a specific area in front of where you're going to actually insert your mutation. And then Sergei's used this approach with allele-specific PCR to demonstrate at a very sensitive level that he's got the correct knock-in here for the 143 and for the 217. Furthermore, he's developed an approach to really optimize doing this knock-in, because while knockouts in zebrafish have actually become almost commonplace, knocking in these specific mutations has really been somewhat challenging. And so what Sergei did is he adapted an approach that's actually been used in human cell lines, where instead of designing your guide RNAs and your oligo to bind on this side, you get them to bind on this side of the genome. And this is this so-called antisense oligo approach. And you can see here for both the 143 and for the 217 in these PCRs, where you look for the knock-in, we get very few knock-ins that are positive compared when we use this sense oligo approach, having it bind here. But when we have it bind here on this strand that's released up when Cas9 does its cutting, we get much more efficient knock-in for both of these mutations, and then that's quantified here as well. And so this approach has been a really powerful approach that we've been able to develop in this project for doing knock-ins of these specific Li-Fraumeni P53 mutations. And then importantly, these zebrafish actually have a phenotype. So I talked to you about looking at apoptosis or lack of apoptosis, cell death. These are live fish live embryos that we stain with acridine orange. Here you see in the wild type, we don't see a lot of staining when they're exposed to DMSO, which is the control. But when we expose them to DNA damage with camptothecin, which is a chemotherapy agent, they get all of these green spots, and those green spots are apoptotic foci. Those are cells that are dying. And you can see that in the R143 TP53 heterozygous larvae, or in the homozygous mutants, you see after exposure to camptothecin, you see much less of this apoptosis. You don't see as many of these green dots. And so this sets up these larvae as a screen that we can use to test for drugs that restore that apoptosis. This is an early sign. These larvae don't yet have cancer, but they have a hallmark that we know is going to make them susceptible and lead to cancer in the future. So one of the opportunities we have with making these P53 fish is we know that in Li-Fraumeni syndrome, patients acquire other mutations that then is what leads to a tumor. And so working together as part of this Terry Fox-funded project, we've been starting to explore some of these modifier mutations that occur in these patients. And this paper came out a couple of years ago by David's group, identifying this microRNA, which is a special kind of RNA that helps regulate how genes function in the cell, that this was associated with patients with Li-Fraumeni syndrome. And so we decided we wanted to look at microRNA 34 in the zebrafish. Again, in zebrafish, it's conserved. MicroRNA 34 is very important. It leads to many downstream functions, and many genes regulate microRNA 34. Here you see P53 upstream. But what we're not sure yet is the relationship really between P53 and microRNA 34. There's sort of conflicting views in the literature. If you test this in vitro or in vivo, if you have loss of miR34A, what that does to mutant P53. miR34A is expressed in the zebrafish. Specifically, you can see here in the brain and along the notochord, and this allows us to do these studies in zebrafish. Again, Sergey used a CRISPR-based technology to, in this case, knock out the gene, and we used an approach called the Multiplex Crispr, where we design a bunch of guide RNAs, and it basically chops the gene up into lots of different pieces so that it doesn't work very well. And evidence for that is seen on this PCR, where this is the wild-type band, and when you chop it up, you get all these different bands because you've got all these little pieces, and so your miR34A isn't working. But what's interesting is, when we look at the miR34A mutants, and Sergey bred these to be both homozygous and created these maternal zygotic mutants, which means there won't be a contribution from the mother that could be providing a little bit of this gene so that it's not completely gone. We completely remove the gene. If you look here with acridine orange staining, as I showed you before, when we treat with DNA damaging agents like camptothecin, we see the same amount of apoptosis. So loss of miR34A in the zebrafish does not lead to any differences in apoptosis compared to the P53 loss or the P53 mutants, where we see absence of apoptosis. Now, importantly, zebrafish have all the same downstream genes from P53 and miR34A, and you can see that here, as Dr. Lane showed you, that you really need to treat with a DNA damaging agent to see induction of these genes. And so when we treat with camptothecin, which are all the red bars, you can see expression of all these various downstream genes like P21 and cyclin G1 go up. In zebrafish, we have not only a miR34A, but also 34B and 34C, and you can see that those genes do increase in fish when we expose them to DNA damage. But we don't see a difference, again, when we have loss of miR34A. We don't see, for example, using P21, we don't see a major difference in P21 levels. And importantly, we do see a loss of miR34A, so that shows you that our knockout is a good knockout. Importantly, we don't see a major increase in miR34B, because sometimes when you have similar genes, you can see an increase in one gene compensate for the other gene. So that's not what's explaining what's going on here. It may just be that miR34 alone doesn't make a difference. And so now what we're doing is crossing these fish into the P53 fish so we can look at their phenotype. Now, as I mentioned to you, the idea of developing these fish is so that we can screen for drugs that restore apoptosis, that restore this DNA damage response as preventative drugs. Because while, as my colleagues have shown you, it's great to be able to treat these tumors and use fish to screen for new drugs to treat tumors, if we could prevent the tumors from forming altogether, that would be great. And these cancer predisposition syndromes are the perfect opportunity because you know that that tumors have the potential to form in the future. And so this is the screen we've done. We can take our various fish, Li-Fraumeni fish, and expose them to drugs. And we can do this in these embryos are really tiny. We can do it in a 96-well plate that's about the size of your palm. And we can expose them to radiation or other causes of DNA damage. And then we can look using acridine orange as a screen. And we're trying to really automate this in a high-throughput way using this special machine called the biosorter, which I'm going to show you a little movie of, is really a flow cytometer for people. So you can see the plate there, you can see that this machine picks up the larvae, and then they go through a series of steps going through tubing. They go to a flow cell, which you'll see in a moment, and that's where the fluorescence is measured, and then that all goes to the computer. So the zebrafish are sort of like a roller coaster that goes flying through this machine. There's the flow cell, and then this all goes to a computer terminal that then reads out what you get, and you sort of can get a result like this. And this is a preliminary result showing you that when you expose to camptothecin in terms of the fluorescence by acridine orange, you can see the wild type have high fluorescence levels. When we use David Langenau's P53 null fish, they have very low levels of fluorescence because they don't have apoptosis, and the Li-Fraumeni mutant is somewhere in between. And so we're working to try to optimize this platform so that we can do this kind of screening in a high throughput way, and we can test hundreds and thousands of drugs a week to be able to find drugs that can restore that cell death. As I mentioned to you before, finding drugs that function is important, but we also have to make sure that they're safe. And this is important for all patients getting chemotherapy, particularly for children who then have a lifetime ahead where we don't want to give them all these secondary problems from our therapy, but even more so for patients with Li-Fraumeni syndrome who potentially could be having multiple rounds of chemotherapy for multiple malignancies. And so many specific side effects of chemotherapy include heart damage from drugs like doxorubicin, anthracyclines. So we can look at heart damage in zebrafish. These are zebrafish that have been exposed to anthracycline. They develop pericardial edema compared to their normal hearts here. We can screen for ototoxicity, so this is the hearing damage you can get from platinum drugs like cisplatin and carboplatin. We can look at these neural mass on the zebrafish surface, which when you bathe them in cisplatin disappear. And so we can look at the drugs, whether that affects these neural mass, which are important for balance in the zebrafish. We can look at their kidney function. So zebrafish, when we inject them with fluorescently labeled inulin, this is the inulin here. Inulin is completely excreted by the kidney, so over time the zebrafish larvae pee out the inulin, and so they're not fluorescent anymore. But if you expose them to a drug like cisplatin that damages their kidneys, you can see the fluorescence is retained because their kidneys don't function as well. They can't void out the inulin, and we can quantify that. So we can use all of these approaches to look for these drugs and see their safety, and so we're really excited to do that going forward. Now, I know our focus is on Li-Fraumeni syndrome, but our lab is interested in looking at other models of cancer predisposition syndromes, and in particular bone marrow failure syndromes. These are diseases where the bone marrow does not function properly, and often these patients then develop myelodysplastic syndromes and acute myeloid leukemia. And this work we've done in collaboration with Yigal Dror, who's based here at SickKids, who runs the Canadian Bone Marrow Failure Registry. And in a paper we published a couple of years ago, we worked together with Yigal to identify a new cause of bone marrow failure syndrome in this gene called PARN, poly-A-specific ribonuclease, where you can see here the zebrafish embryos that have knockdown of PARN, that don't express PARN, are anemic. This is O-dianosine staining looking at hemoglobin. They don't make hemoglobin, and many of the genes that are involved in controlling red blood cell development like GATA1 and white blood cell development like MPEG and MPX, those levels are decreased. And so more recently, we've been embarked on a new project with Yigal looking at Shwachman-Diamond syndrome, a bone marrow failure syndrome associated with low white blood counts, but also these other features and a predisposition to MDS, myelodysplastic syndrome, and AML. Most patients have a mutation in the Shwachman-Diamond gene, and this gene's involved in ribosome biogenesis, making the ribosome that's important for making mRNA and protein translation. But what's important, particularly for this audience, is the secondary mutations that often cause leukemia in patients with Shwachman-Diamond syndrome are actually somatic mutations in P53. So you've heard a bit about that already today. We think of Li-Fraumeni syndrome where P53 is the first hit in the genome, and then you get other mutations. But in other diseases, you may have one mutation, then P53 is the second hit. And that was shown here by this paper in the New England Journal last year, where they found the number of patients that had TP53 mutations was quite high in patients specifically that had Shwachman-Diamond syndrome, and that those patients that had Shwachman-Diamond syndrome and developed MDS or AML really had some of the worst outcome of any of these patients with MDS and AML. So something worth studying. And so Yigal's group, along with another group, recently discovered a new cause of Shwachman-Diamond syndrome called DNA-JC21, which is, so these are patients that had this disease, but they don't have mutations in the classic SBDS gene. And one of these patients actually developed acute myeloid leukemia. This gene, we don't know exactly what it does, but from work on the homolog in yeast, it also seems to be involved with the ribosome, specifically the pre-60S ribosome, and packaging it properly. And here you can see the different mutations that have been identified. And so we're in the process of making zebrafish models of this disease, here knocking out the gene initially with morpholinos, and you can see this is a whole mountain situ hybridization of zebrafish larvae, and you can see here are the wild-type fish that have all of these purple myeloid cells that are much greatly reduced in the in fish lacking DNA JC21, and now we're using CRISPR technology to make germline mutants of this DNA JC21, and we will cross these with the, these are David Langenau's P53 talonfish that you can see develop tumors and see what phenotypes we get. So in summary, I hope I've been able to show you that zebrafish really are a robust animal model for studying Li-Fraumeni syndrome and modifier mutations that may lead to cancer progression, that we can use this to screen for drugs that actually prevent cancer development, and that we can then look at other diseases that may involve P53 as a secondary hit that lead to progression of cancer in predisposition syndromes like bone marrow failure. Some people have a trump card. This is my trump fish. And a lot of groans, and most importantly, I want to acknowledge my group, and specifically those people that I've highlighted here are the ones who have done some of the preliminary work. My collaborators, particularly David and Adam and Yigal for the bone marrow failure work, and our funders, and I'll be happy to take questions.
Case there's any burning questions in the fish.
So we learn all of a sudden that the zebra becomes a main player in the arena of the Li-Fraumeni. I wanted to know, do you really anticipate that you are going to prevent the development of tumors with these models, or use them as a model for testing the efficiency of therapy?
So that's
because
a great.
These are two different.
Sure. So the question is with the ability to use the fish for both either prevention and or to test sort of the efficacy of therapy. And I think our hope is that we'll be able to do both. There are a number of drugs now that are P53 restoring agents that people have looked at that we may be able to use, look in these specific different mutational backgrounds, which of those work better or not. Again, we think that these are the fidelity of these mutants to the specific specific mutation in given patients is pretty good. But ideally, it would be nice to be able to see, could we find drugs like the Gleevec of Li-Fraumeni syndrome? That, Gleevec is a drug that we give in CML for patients that are identified to have the Philadelphia chromosome. We give it long term, and it prevents sort of the evolution of that disease. Maybe we have the opportunity to find drugs that similarly either restore P53 or prevent the oncogene function of mutant P53 that we can use almost as a cancer controlling agent. So I don't think that's off the table.